Good morning, everybody. My name is Daniele Conficoni. I am a doctor in veterinary medicine and I have a PhD in veterinary science, and I come from the University of Padua. Today I'm going to show you something about my topic. Today we will start from the basics, and we are going to talk about bacteria and their behavior. Today we are going to see behavior and the physiology of microorganisms. In particular, as you can see from the picture, of eukaryotes and prokaryotes. So first of all, we will start with some classification hints, so some information about classification, and then we are going to compare eukaryotes and prokaryotes, and then we will see also some viruses. The classification is a way to understand the order of the distance between the living beings. In particular, with this classification we are able to highlight the phylogenetic hierarchy between living beings. And nowadays we do this classification with DNA hybridization and RNA sequencing. So in this way each living being could be classified with subsequent groups. So we have the domain, that is the widest group, then we have the kingdom, then the phylum, that is smaller, the class, the order, the family, the genus, and the species. With genus and species, we are able to identify one specific species of animals. As you can see, we have the time has split, so we started from a common ancestor that developed the bacteria, the archaea, and then the eukaryota. This is a phylogenetic tree, and highlighted the distances between different living beings. So we have the big group of the bacteria, the group of the archaea, that are close to both bacteria and the eukaryota, and then the eukaryota. Here we have some bacteria, as you can see the gram-positives, of which we are going to talk about. But also in the eukaryotes we have some animals, the fungi, plants, and so on. The classification, as I said before, starts very wide, so we have the domain and so on. In this case, we have a red fox, that is an animal that is classified as it. So we start from the domain, that is eukarya. So the fox is a eukaryote, so it is in the domain eukarya. It is an animal, so it is in the kingdom Animalia. And then the phylum is the chordata, so the animals with a spine. The class is Mammalia, so animals that will give birth to their offsprings and they will milk them and something like that. Then we have the order, Carnivora, so meat eating. And we have the family, Canidae, in which we would have also dogs and wolves. Inside the families we have the genus, in this case is Vulpes, and then the species, that is Vulpes as well. Here we have the comparison between a eukaryote in green and a prokaryote in red. The big differences between the two types of living being are mainly due to the complexity of the eukaryotes respect to the prokaryotes. In fact, eukaryotes have a lot of organelles inside them, so they have the nucleus with the genomic material, and then they have also other stuff. While the prokaryotes inside are a little bit of a mess, and they have the ribosomes like the eukaryotes, but they are just in the medium. The big differences are these ones, so the dimension. Eukaryotes could be 10 times bigger than prokaryotes. Then the nucleus, that is absent in prokaryotes but present in the eukaryotes. The number of chromosomes. Prokaryotes have always only one chromosome, while eukaryotes usually have more than one. Ribosomes, so the dimension of the ribosomes, that are the protein that will translate proteins from mRNA, they are bigger in eukaryotes and smaller in prokaryotes. The peptidoglycans, that are a kind of molecule, is present only in the prokaryotes and it is absent in the eukaryotes. The flagella that are used for the movement are different. So the prokaryotes have a rigid flagella made of flagellin, while the eukaryotes have flagella made of microtubules that will give more flexible and not rigid flagella to the eukaryotes. So the actual dimension is very different. So eukaryotes are very big, while prokaryotes are very small. And prokaryotes could affect eukaryotes. Okay? So now we are going to see some small hints about the domains and the kingdoms. So the eukaryotes, so the domain Eukarya, they are divided in 4 kingdoms. And the kingdoms are Protista, Fungi, Animalia, and Plante. So the Protista are unicellular eukaryotes. They are simple eukaryotes. They are mainly algae and protozoa. They are autotroph, heterotroph, and intracellular parasites. So they could be they could have different nutritional behaviors. Then we have the kingdom fungi. The kingdom fungi is composed of yeast, molds, and mushrooms, and so on. They absorb the organic matter through their plasma membrane, and they could also give some problems to human, for example. And then we have the kingdom Animalia, that is composed of multicellular animals. They ingest organic food through a mouth, that is really important, and the cells are organized in tissues, and the tissues are organized in organs, and the organs in systems, and the systems will create the living being. The last kingdom of the domain Eukarya is the kingdom of Plante, of multicellular plants. They are able to photosynthesize and to convert CO2 and water into organic molecules, and they are organized as well as animals into tissues, organs, and systems. The group of prokaryotes is divided into 2 domains, okay? So the domain bacteria and the domain archaea. The domain bacteria have peptidoglycan in cell walls. It comprises all the pathogenic prokaryotes, many non-pathogenic, but also all the photoautotrophic prokaryotes. The domain archaea are all prokaryotes with cell walls without peptidoglycans. And they usually have unusual metabolism, and they live in extreme environments. They are similar to both bacteria and the eukaryotes. And as we saw in the phylogenetic tree, we had the splitting between bacteria and archaea, and then from that splitted archaea and the eukaryotes. But what about viruses? Viruses are different because they are not classified as a living being. They are acellular, so they do not fit in any domain. They are obligate intracellular parasites, and they are evolved to infect cells because they lack the components that are able to multiply them. So their replication could happen only inside a host. Usually they only infect one specific type of cell because they are adapted to that specific type of cell. And they are very specific to their niche. They are very small, so even smaller than prokaryotes, and they range in the size between 20 nanometers and 500 nanometers. And only in some cases they are bigger. All the living beings could be affected by viruses, so by viral infection. They are composed of different parts. Their outside is composed of protein shell that is known as capsid, that is usually simple, very simple, because it is usually constituted of the many repeats of the same protein unit, because inside the virus could be just be a very small fraction of genomic material. So if the capsid is made of the repeat of the same protein unit, the genomic material could be smaller. So inside the capsid we have for sure the genomic material and maybe some enzyme, and this capsid could be symmetric or asymmetric. The genomic material of the viruses could be DNA or RNA. So two different types of genomic material, and this division could be also inside each one, so it could be single-stranded, so just one strand, or double-stranded, like DNA. And both RNA and DNA could be single or double-stranded. In this picture we can see different type of symmetry, so we have helical symmetry, polyhedral, spherical, or also complex. The last is the bacteriophage, that is a virus that is able to infect bacteria. So bacteriophage, from Greek, bacterio is bacterium, phages means able to eat. So here we have the classic division of viruses. So we have the first division is the type of genomic material, RNA or DNA. Then we have the symmetry. Then we have the naked or enveloped. So if outside of the capsid it has or not a plasma membrane. Then we have the dimension and so the name of the family. But let's talk about bacteria. Bacteria, based on their cell wall, could be gram-positive, gram-negative, or also exist the so-called mycoplasma that have no cell wall. In bacteria we have some spore-forming species that are present only in gram-positive bacteria. They could be able to produce biofilm, and they have one circular chromosome. They could have different shapes, so cocci, bacilli, and sometimes spirochetes or curved rods. They move again with the flagellum and they attach to other cells with the pili, called fimbriae. Here we have the genetic distances between the bacteria. In the upper part we have Campylobacter, so proteobacteria. Then we have the firmicutes with the gram-positive, and then we have all the other bacteria. So here are the shapes. As you can see, we have the diplococci, the streptococci, so one after the other, the staphylococci that have an arrangement that is a complex arrangement. Then we have the spores, and then we have the spirals and the spirochetes. But what is the difference between gram-positive and gram-negative bacteria? It's the cell wall composition. So in gram-positive bacteria we have a very thick wall of peptidoglycans, while in gram-negative bacteria we have a small wall of peptidoglycans, and outside of it we have another plasma membrane. And because of this division, They are called gram-positive or gram-negative because with the staining, with the gram staining, they will stain with different colors. So here we have the Staphylococcus aureus, that is the gram-positive cocci, that is in purple. So the cocci in purple is the Staphylococcus, while Escherichia coli, that is a gram-negative, that is a rod, a bacillus-like, they are in red, in pink in the picture. So this is how, after the gram staining, we will have the different bacteria. Now, some hints on spore formation. So inside the cell there will be a multiplication of the DNA, and then after the replication the cell will divide and will create a mother and a prespore. The prespore will form at the end the spore. So after the first multiplication of the DNA, this prespore will be just coated by different piece of coating, and at the end they will form a free spore. The spores are important for us because they are very resistant in the environment, and they are very hardly eliminated by classical treatments. Now we are going to talk about yeast and molds. So fungi, they are eukaryotes. They are important for the production of fermented products, so like cheeses, but also some kind of alcoholic beverage and so on. They could spoil, usually they are mediator of spoilage in food, and they could be an hazard only if high quantity, and especially for people at high risk, the so-called YOPs. YOPs means young, old, pregnant and immunocompromised people. They could produce also some toxins, the mycotoxins that are toxin for human, but also antibiotics. So some molds are able to create antibiotics like penicillium, and because they use these toxins to don't have competitors in the substratum. They are different. So yeast usually are reproduct asexually with the mitosis, with the budding. So from one piece, just one other piece will be created. They are used for the production of ethanol in alcoholic beverages like beer, and they are leavening agents for baking. They are also could be nutritional supplements for vegans. And the molds are more complex. Their reproduction could be sexual, but also asexual with the production of small spores, but also with the mitosis. They are used for the production of some drinks like soy sauce, sake, corn, cheese, some salami, and both of them are important for our technologies and for our food products. Now we are going to see some growth factors in food. So very quickly we are going to see the intrinsic factors, so physical and chemical food properties, the extrinsic factors that are related to the environment in which the food is stored, the implicit factors that are related to the microorganism itself and their interaction in the food. Then we are going to have the processing factors that are factors influencing the food, so how the food is processed, and then we are going to quickly see the combination of their effect, the so-called the Arnold concept. So first of all, the intrinsic factors. The intrinsic factors are that values that depends on the food, so the water activity, the hydrogen ion concentration, so the pH, the nutrient content, so what is inside it, then the redox potential, that is how easily the substance loses electrons and becomes oxidized, or gains electrons and becomes reduced. Said with a simpler word is how much oxygen is available inside the substrate. The antimicrobial substances and mechanical barrier to microbial invasion and their biological structure. So now we are going to quickly see almost all of them. So the water activity is the moisture content of food. It's important, so it's the free water inside food. It is important because if it is very low, bacteria could not survive in it. Usually it's very high for fresh products and very low for not fresh products. And each organism has a maximum, an optimum and a minimum for their growth. Usually bacteria need higher water activity than yeast and molds. That's the reason why only yeast and molds are able to spoil some kind of very dry food. So foods could be divided in high moisture, intermediate moisture and low moisture food, and let's say that the majority of fresh products are high moisture food, so with a water activity higher than 0.85. Here we have the majority of them, so fresh vegetable, meat, milk and fish, they are between 1 of water activity, so the maximum, so pure, like pure water, and 0.95. Here we have the water activity in the food, so fresh products have a lot of water, free water, while dried food have very low amount of it. As you can see here, especially meat, vegetables, meat, they are like a bag of water with some proteins, some amino acids, some sugars, and so on, some lipids inside. So when we think about meat, like a piece of meat, we are thinking about a water bag. So based on the minimum requirement, bacteria could be divided in different kind of bacteria, but the bacteria important for food safety could survive with only high water activity. So like Pseudomonas aeruginosa that will spoil our food, but also some pathogens like Escherichia coli, Clostridium botulinum, Salmonella, Bacillus cereus. The pH. Now let's talk quickly about the pH, the acidity of the food. The neutrality is at 7, higher is basic, lower is acidic. Based on this value, bacteria could survive or not. The more close to neutrality, the more they are able to survive. Usually bacteria have a shorter range of growing, while yeast and molds have a wider range. Again, the majority of bacteria could survive and grow with a not so low water activity pH, but a lower pH will inhibit them. In particular, like Pseudomonas 5.6, while as you can see, the yeast and the molds, they are very low. They could grow with a very low pH. According to their pH, food could be classified as low acid foods, or medium, or acid, or high acid foods. So there is a wide misunderstanding, and according to the normal people, let's say, the food are classified with strange values of pH, but in particular we could say that fruits usually have a low pH, like apples that are 2.9, 3.4, limes 1.8 and 2, while vegetables could be close to neutrality, like lettuce, peppers, or with a lower water activity pH, like tomatoes. Meat have usually a higher pH, close to the neutrality, as you can see beef 5.1, 6.2, but also fishes are even closer to neutrality. The dairy products as well could be a little bit more acidic based on their type, especially yogurt have a lower pH. So nutrient content, very quickly, there are the contents, so based on the contents of the nutrients available, different bacteria could survive or not. The redox potential is the amount of oxygen, of free oxygen in the food, and we are going to stop here, and this redox potential will allow a bacterium to grow or not, and a solid food usually have a lower redox potential, while a ground food like meat have a higher redox potential because it will be inside oxygen. So thank you for listening, and see you in the next lecture maybe.